Is my screen visible?
Yeah, your screen is visible.
So today we will talk about the quality control and trimming. I just sent some GitHub links in the last class, the GitHub URL that is. I wanted you to have those files actually. So these are this one and this one. It is actually paired in file from the NCBI. So I hope you have this file, but you don't have. We just nothing to worry about that. So quality control actually ensures that our file has not any low quality basis or the adapter contents. These are the two main things, that is low quality basis and the adapter contents. So today we will assess the read quality using the FastQ tool. Then we will trim our adapter contents and the low quality basis using the trimmometer, and we will try to understand how this thing happens. And I just told you we will use these two files. So we will conduct in single mode, single in mode, and also the paired in mode. So before starting, let's understand a little bit about the FastQ format. I hope you understand it already. So every FastQ file, every FastQ read must have 4 lines. So the first line must contain the at the rate symbol, and after that the identifier of the read, that's called the ID. Then our sequence, that is the read. Then plus symbol, it's used for the separator, and the fourth line is quality. So here we can see a sample FastQ file. So when we look at a FastQ file, what we see actually here for this sequence, for this read, we can see poly a tail. For the trimming part or for the mapping part, when we will proceed the mapping in the third lecture, for the mapping part, we must need to trim poly a tail, poly G tail, and okay. If we look at here, the picture is missing. Just let me. So when you can see the read here, that is the quality scores. These actually the quality scores, and it is basically in ASCII character. And these are this line also. And in ASCII, there are different meaning for different characters. And the question comes: all the ASCII character, that is ASCII character usually 0 to 127. Does all the ASCII character are used for the quality scores? The question is no. Sorry, the answer is no. So we can see this table. This is one column, this is another, this is another, and this is another. And for the PH rate 33, when we talk about the quality score, there are two actually: PH rate 64 and PH rate 33. PH rate 64 actually for the old version of Illumina, and for the modern version, the PH rate 33 is used, and it actually starts from the ASCII character 33 and ends at the 126. So in ASCII character 33 to 126, so these 94 bases or these 94 characters are used for the quality scores. That is the PH rate 33, and it actually is the mapping PH rate 33. It actually called the MAPQ. That is the mapping quality. And first one is exclamatory sign, and here you can see the hash sign. So for the hash sign, the mapping quality score is 2, and we see the I sign. For the I sign, the mapping quality score is 4. Okay, so we can go here. Mapping quality 40. This means these are very high. So how we know that these are very high? Here are some formula to calculate the mapping quality and the accuracy from that. Suppose we have a mapping quality 40, that is the quality score 40, and for that, if we place a score in here in this equation, we will get this one. This is the error rate. So, and from the error rate, we can calculate the accuracy. That is, if we subtract the error from 1, we will get the accuracy. So this is the accuracy, and it actually 99.9% accurate. So when the mapping quality is high, that means it has chance that the accurate it actually how much accurate or how much probable the accuracy this read has. So if we look at mapping quality 2 and we place a value in here, we get this. So it has chance 63% error, and it has the accuracy rate 36.90%. So we expect the high mapping quality. And here you can see the hash sign. We just saw the hash sign has the mapping quality 2. We don't expect that. We must trim the n basis, and it usually has the low mapping quality. And here we can see poly A chain, and it has also the low quality. This read contains the low mapping quality at the first, and it has a short read actually, and most of the read has the low mapping quality. So this is the how we can if we look at the FastQ file, what we can see actually, we can see the poly A tail or poly G tail. We can see the low quality scores, or if there are any n bases. And if we look at a simple example. So we are dealing with these two FASTQ file, read1, and this is the read2. If we open the if we read the first 3 reads from each file, so you can say why I am using 12 in here. I just already told you a FASTQ file must contain 4 lines for one read. So I expected 3 reads, so it will be 3 into 4, that is the 12. And if I read the second R2 in here, so just look at the position. That is for the for the first read in R1 and first read in the R. The identifier are actually same. So for all paired in file, we must have the corresponding read in the same position. That is, if we think this as position 1, so what identifier it has, the reverse complement should be in the same position in the R. It actually means while it's sequenced, when we take a DNA fragment, the DNA fragment sequenced in forward mode, that is right in the R1 file, and it sequenced in the reverse mode, it right in the R. So FASTQ file read1, sorry, R1 and R2 must have the same number of reads. How we can confirm that? Simply we can use the WC command. Just give the FASTQ file name as in, and we can pass this command using a pipe sign. So for this FASTQ file, we have this number of reads, and for the second one, we can check. So you can match the read number here and here. So these are the same. I think something is clear. That is, for R1, we must have the corresponding read in the R2, and both R1 and R2 must have the same number of read. If we go back into slide, so now we can proceed for the quality control. So how it works actually. So for the FASTQ file, this is the command FASTQ, and for we can specify the output directory. We can specify the thread, that is FastQC support multi-threading or parallelization, and we need to pass the read. So if we go for FastQC, we are specifying the directory. Okay, I need to show you what this directory actually contains. So I already created a directory QC, and we have the read in the data folder. We need to pass our out into the .qc. Dot means the current directory, and the QC for QC directory. If the file size is small, then we don't need to specify the thread command. Otherwise, we can use it. And then our file name. So inside data, here we can specify as many file as we want. So we have two reads, that is R1 and R2, and I am just passing the two file names in here. And if we run it, then we need to take the output in the QC folder. So if we open the QC folder in here, we just generated this one and this one.